Hi everyone, my name is Karen and today I'm going to present to you all choline availability and how it alters embryonic development of the hippocampus and septum in the rat. So, a bit of an introduction. As we know, choline is an essential nutrient for humans and rats. Um, Dietary choline availability during pregnancy can have a long-lasting behavioral effect in offspring. Prenatal choline supplementation in rats often promote cognitive function and visuospatial memory. So during the fetal brain development, progenitor-type cells in the ventricular neuroepithelial layer of the hippocampus and septum undergo cell proliferation and mitosis. So, specialized regions of the hippocampus, such as the fimbria, dentate, gyrus, and the Ammon's horns, these help develop. So, these develop from the progenitor cells, and they are associated with the different aspects of memory and learning processes associated with the hippocampus. So, neuronal patterning and brain function should be significantly influenced by alterations of timing of cell division, migration, and apoptosis during development of regions. So materials and methods. So one of the methods that they use is timing the breeding of the animals. So they bred them. 8 to 10 days later, they would be maintained in a climate controlled environment, and they would be exposed to light and darkness for 12 hours. So most likely sleep 12 hours, and they were active out and about for 12 hours in the sunlight or in a lighted room. So the rats were provided with AIN 76A diet, which contains 7.8 millimoles of choline chloride and as much water as they want. So day 12 of gestation, pregnant rats were divided into 3 treatment groups: supplemented, control, and deficient. The controlled group was given AIN 76A diet with the choline and water, and it was sweetened. The water was sweetened with 50 micromoles, I think that's what it is, of saccharin. So then the supplemented rats were given the AIN 76A diet, but the water was supplemented with 25 micromoles of choline chloride, and it was sweetened with 50 micromoles of saccharin, the sweetener. The deficient group, they were basically not given any kind of choline, just to see what happens. So the treatment continued until gestational day 20. On embryonic day 18 and 20, animals from all treatment groups were anesthetized by injection of sodium pentorbital, so 1 milliliter per 100 grams of body weight. So assessment of apoptosis, combination of apoptotic morphology and a tunnel were used to detect apoptotic cells in the fetal brain, and then assessment of mitosis. So it was determining if mitosis was altered by the chlorine availability in the maternal diet. All right, so the results. So neuroanatomy, so day 18 in controlled brain, neuroepithelial layer and central region, so CA1 and CA3, are identifiable, and cells migrating from the overlying neuroepithelium have begun to portray circular outline of dentate gyrus, So then in day 20, neuroepithelial layer adjacent to septum slowly began to decrease in thickness. So little change in hippocampal structure from day 18. So there was basically not much of a difference during these days. So then mitosis, a fixed length of the neuroepithelial layer was examined beginning at the junction of neuroepithelium and corplex, extending towards the cortical neuroepithelium. Measurements of accumulation of mitosis were taken. So on day 18, smaller portion of cells were undergoing mitosis in CD compared to CT and CS in brain. So then day 20, neuroepithelium in CS brain had increased rate of mitosis compared to control and CD groups. So in this area, in the apoptosis, in day 18, the controlled group and the supplementation group, they were basically almost identical. There's like a slight difference, but it's almost the same. But as for the deficient group, it was a lot higher. So this basically just shows that when you're choline deficient, apoptosis basically increases. So in the fimbrial glioepithelium and fimbria, choline deficient decreased the number of cells born on day 16, then accumulating on day 18 compared to the controlled and the supplemented. So then choline supplementation increased the number of cells born on day 16, present on day 18 in the fimbrial glioepithelium compared to the controlled group and the deficient group. So results. Choline availability during pregnancy affects development of hippocampus and septum in fetal rat brain. So feeding choline deficient diet during embryonic days 12 and 18 reduced mitosis and birth of cell and progenitor neuroepithelium ventricular zone in day 18. So choline deficient, as I said before, it actually increased apoptosis in specific regions of the hippocampus. So when feeding choline supplemented diet, it results in an increased number of developing septum. So with availability of choline in the diet, it can be critical for timing patterns of mitosis and migration of cells developing hippocampus and septum. So apoptosis and cell proliferation are two interrelated processes that regulate cell number during fetal brain development. So this study shows that when there is a decrease in choline during the 12 to 8 window of brain development, it leads to an increase of apoptosis that is focused primarily in the dentate gyrus. So the results suggest that the effects of choline availability on cell proliferation, migration, and commitment to differentiation are significant for brain development than in apoptosis during this period of gestation. So dietary choline may affect the stable neuronal population of the hippocampal formation and other brain structures through mechanisms related to cell death and to the complex events associated with migration and differentiation. So